Good morning, everybody. Or is it afternoon? Not quite. I think it's morning still, isn't it? Thank you for joining us today for this PestExtra live presentation. We have Alan Buckle today from the Rodenticide Resistant Action Group UK. He'll be talking about 60 years of anticoagulant resistance in the UK. But just before we get started, I'm going to do a few housekeeping items. Some of you may have been through this if you've been on some other seminars today, but just to get through it for those that are new. CPD, the PestExtra event team, will be tracking each individual's engagement and activity across all areas. So it's a little bit different to before. So, for example, any presentations you join or any videos you may be watching, or when you go and see the exhibitors on these stands, which you must do, they've helped to set this up today, and they're a really integral part of this PestExtra. So make sure you go and see them on their stand. This couldn't have happened without them. Any of those things that you get involved with or anybody you talk to, we'll be tracking that on your personal account. We'll add up what hours you spent with us, and each hour will add 1 CPD point. So if you've been with us 4 hours, you'll get 4 CPD points. So hopefully that makes sense. If you want any more information, just go to the BPCA stand back in the main lobby area. We also have a Q&A section for you to ask questions to the presenter. We'll do this at the end. But you'll see an option to click on a Q&A and submit your question button. And at the end of the presentation, I'll read these out for Alan to answer, and yeah, we can get them answered live. If we do run out of time, because he's very strict on time, we need to be allocated within the 45 minutes that we've got. Then make a note of your question in your notes section, and then by all means, if it's something you really need to get answered, pop it over to the BPCA team or go onto the BPCA stand, and we'll try and get it answered for you. Also, if you see a question that pops up, now all the questions that you ask and that you type into the Q&A section will be visible to all of the other attendees. So if you see a question on there and you think that is a brilliant question, then give it a thumbs up, because then I'll be able to see that if we've got some questions that a lot of people are interested in, I'll make sure I get those ones answered first. Okay, so keep an eye on that. We also have a discussion forum. So if you just want to have a chat to everybody, say hello, maybe a few comments, but it's not a specific question, we've got that discussion forum there. The BPCA moderator will also be on there, so they'll be able to pick up any problems that might be occurring. In terms of technical problems, hopefully no one will have any, but if you do have any sound or video issues, then there is an orange live support button on your screen. It should be quite clear. So technical problems, click on that orange live support, okay? And in these live sessions, we can't hear you and we can't see you. If you do want to have some live chats with the BPCA team or our exhibitors, then please, give us a visit on the stand. That would be amazing. Great. Well, that's me done. Alan, let's get over to the most important bit, your presentation. So please, I look forward to it. Thank you.
Okay, Natalie, thank you very much for the introduction. Well, I hope you can all see me and you can see my screen. I can't see anything other than my screen, so I hope it's good. Just a couple of words on this on the title slide. I set off to do this presentation and I got to slide, I think 21 it was, and I hadn't finished Norway rats, and I realized that there was no way I was going to be able to cover both Norway rats and house mice in this presentation, so I've just stuck to Norway rats. If there are some questions on mice, we can take those at the end, but apologies, you're going to have another presentation from me if you want to hear about the house mouse story. The other thing I should say, I'm the former chairman of the RAG. After doing that job for 11 years, I've handed over to Matthew Davis of Kilgerman, and he's the new chair. So it's 60 years, because I'm sure you know, 59, some say 58, 59, 1959 it was where the very first incident of anticoagulant resistance popped up in Scotland, and we'll be saying a bit more about that on some of the slides. So I'm going to move on to the first slide. There are just a few sort of introductory slides so that you understand some of the terminology and some of the words that I'm going to be using through this. So just a couple of intro slides. So the first thing I wanted to say is about resistance. What is resistance? And it means lots of different things to different people. And I've just put 4 meanings up here, and there are some others, but the first one we often hear about is natural resistance, and that is that some species are naturally much less susceptible to anticoagulants than others. And for example, the house mouse is much less susceptible to anticoagulants than the Norway rat is. So it means they need to eat more on a weight for weight basis to get a lethal dose. There's metabolic resistance, and we're learning more and more about this. Some animals have an enhanced enzyme system that enables them to get rid of foreign chemicals, and amongst those, anticoagulants. You've all probably come across behavioural resistance, and what we have is that some populations learn to avoid baits or even the apparatus of bait application. Excuse me. And there's finally the main one, and this is the one that I'm going to be talking about for the rest of the talk, really. This is genetic or molecular resistance, and this is when a mutation happens in the DNA sequence. That sequence modifies the manufacture of an enzyme called the VKRC1 enzyme, and the modified enzyme, anticoagulants find it harder to bind with, and therefore animals that have that modified enzyme are said to be resistant. I'll say much more about that. But these 4 different types of resistance are really not mutually exclusive. One animal can have 1, 2, 3, or even 4 of them. And I'm pretty sure that some of the Norway rat populations that we deal with down here in the south of England have certainly got genetic molecular resistance, because we can find that in their DNA. I'm sure they've also got behavioural resistance, because we find them very hard to bait, and those same populations also probably have metabolic resistance as well. There's been some new studies on the central southern England resistant rats to show that. So you have to think that the population that you're dealing with may have one of these, and we often just think about this bottom one, the genetic one, but they may well have these other resistances as well, and house mice certainly all have that top one. Okay, now this is a picture of that enzyme that I was telling you about. I just want to explain a few bits about the structure of this enzyme. It's like a string of beads. You can see the string of beads, and each of the beads is an amino acid. And if you see, they're counted from the bottom left up to the top right. They go from 1 to 160, 1, 2, 3. So there's 163 amino acids in this string. It's a membrane-bound enzyme. In other words, it sits across the membrane of the endoplasmic reticulum in the liver cells of us, as well as rats. And as it passes through, it goes through 3 times. And in 2 of those passes, that's where the business gets done of the enzyme affecting the vitamin K cycle. And you can see in the colors down there, and you can number those amino acids down to 120. You can see the green ring. And then 128 is the L, then 139, and so on. And those are the specific amino acids that change in the mutation. And you can see they're all in these what we call redox centers, or the actual reaction center where the enzyme does its work. So just single changes of those single amino acids makes all the difference for the way this enzyme works. And what I'm going to show you now, there are 5 main rat anticoagulant resistance mutations. There are more than 5, but there are 5 that we know affect the efficacy of anticoagulants. And in this slide, you can see where we found them first. There's Scotland, Wales, Hampshire, Berkshire, Kent. And then the last one, we found it in lots of places, but mainly in Yorkshire first. And you can see there in green, that's the amino acid that the enzyme should have. And then the place where the change takes place, that's a point 128 on that string of beads. And then you can see the mutated enzyme, the mutated amino acid, which in that first case it goes from leucine to glutamine. Now I'm going to be using these short names down the side a lot in this presentation. So there are 5 of them to think about. Now, the L stands for leucine. Unfortunately, when they got to glutamine, the G had already been used up as an abbreviation, and they had to give it a Q. So we talk about L128Q, and when they got to T for tyrosine, the T's had all been used up, and so they gave that a Y. So we talk about Y139S for the Welsh resistance. So I will be using those short mutation names quite a lot through the presentation. So just one other bit of terminology, if you like. I'm going to be saying the words heterozygous and homozygous, and I suspect you know what this means, but some of you may not. Everyone's genetic material is carried on chromosomes as two copies. You'll remember the DNA helix is like two spirals interlinked, and one of those spirals comes from mum and one comes from dad. And down each of those spirals, you get one copy of each gene. So everyone's got two copies, one from mum and one from dad. So where the two sets are different, in other words, a different gene was obtained from mum and a different one from dad, we say animals are heterozygous. They've got two different copies of the gene. And in our case, one of those is resistant and one is susceptible. Where the two sets are the same for a specific gene, we say that animal is homozygous. So when an animal is homozygous for a resistance gene, it's received that mutation both from mum and dad. And usually it means if there's a high level of homozygosity, it means there's been lots of selection pressure. It means lots of resistant animals have been surviving, mating with more resistant animals and building up that population where each has got a copy of the gene, both from mum and dad. And homozygous animals are usually more resistant than heterozygous animals carrying the same gene or the same mutation. Okay, so I want to go back to the start, and it's about the only old slide that I've got here. But this is pretty amazing if you think back. So this is the result. It was published in '73 of some surveys that were done between '59 and 1970 of what was known then as warfarin resistance. We only knew about warfarin resistance in those days, and in all of these places, you can see on the map. At least I hope you can see. Those of you on the phone, some of these maps may look a bit difficult to see. I apologize for that. You will be able to have another look at it on another device if you wish later. But all of those spots are resistances. And I'm pretty sure when you look at where those spots are found by 1970, all of our 5 resistance mutations were already around at that very early stage. So what we think is many of the current second generation anticoagulant resistance foci were already present, even though eschars hadn't even been invented at that stage. So in those days, so-called warfarin resistance was actually multiple different types of resistance. And eschar resistance was being selected for by warfarin and the first generation anticoagulants because the eschars just weren't around in 1970. So eschars selected for future FGARs selected for future eschar resistance. Probably all 5 RAC mutations were around, and the use of eschars did not cause resistance, but the way we've used them since probably helped it to spread. I want you to keep that that map in mind. You can see a big resistance focus in Wales on the Welsh border going right the way over to the coast. You can see a big focus down in Kent and East Sussex. Obviously, lots of resistant rats there in those 2 big major foci. You can see up in Scotland, in the central belt of Scotland, another quite large area of resistance. And those 3 foci are still there, and we've got lots more. So I'm going to start talking about the different resistances. And on this first map, what I'm showing you is all of the fully susceptible animals that we've come across while we've been doing the resistance DNA sampling. So these are animals that got a susceptible gene both from mum and from dad, and so they're fully susceptible. And you can see in the UK, susceptibility is very widely dispersed. So there's still lots of susceptibility out there. But some areas we've done lots and lots of testing, and you'll see this in the later maps. So down in the central southern England, we've done masses and masses of testing, hundreds of rats we've looked at. And those few spots down there are the only susceptible animals that we've seen. So down in that central southern England area, there's very little susceptibility actually. Otherwise, it's pretty much spread around. There's been very little testing in many of these places, and I'm going to tell you where we need lots more samples from a bit later on. So if there's not much testing, we haven't found much resistance, we haven't found much susceptibility. It's not surprising to find susceptibility around because even when two animals each have one copy of the gene, so they're heterozygous, if these two heterozygous animals mate, 25% of the offspring will be fully susceptible. So it's not surprising that we're finding these susceptible animals. That little spot right out there, if you can see that, right out in Pembrokeshire, a seagull dropped a rat on my head in the garden in the house I was staying in out there. I didn't realise where it come from. It just landed in the garden. The seagull came down and tried to get the rat, and I beat it off, chopped the tail off, sent the tail to Reading, and that's how we got a little spot right out in the far end of Wales. But you can see we need lots and lots more samples from many places in the UK. So this is the first resistance. This is what we call L128Q, first resistance ever found in Scotland. It's got strong resistance to all of the FGARs, but it's fully susceptible to all of the SGARs. Now you can see where it's spread to, where we've been finding it quite a lot in Greater Manchester, a bit in West Yorkshire. It looks as though it started up in Scotland. It probably came down the A1, and so it's in Tyneside and Durham, probably over the Pennines, and landed down in Manchester. But it really hasn't spread that much. We don't get much of this, and it's probably because all of the SGARs are fully effective. So when there was a time, some of you may remember, when we couldn't use flocoumafen diphthialone for rat control because of the indoor-only restriction, that didn't matter with this resistance. And so it's been pretty localized. Now, later on, I'll show you what's happened with the very severe Hampshire resistance that that bromadiolone and difenacoum, which we could use outdoors, doesn't work against. And you can see what's happened with that one. So here's the next one. This was the next big resistant focus that was found. You saw it on the first map, found in 59. It was once the largest known focus. These days, we don't get many rats sent from there for DNA. I'm quite sure the area of resistance is much bigger than is shown in those little spots that are in Shropshire and just on the Welsh border, where a good friend of mine, Ewan Bates, regularly sends us rat tails from there, but nobody else does. And so we really need lots more samples from there. So it's got very strong resistance to FGARs, full susceptibility to the potent SGARs, brodifacoum, diphthialone, and flocoumafen, some lowered susceptibility to bromadiolone and difenacoum. Well, for 50 years, it was quite astonishing. We found it nowhere else. And then quite suddenly, a couple of years ago, we started to find it just to the northwest of the city of York. And it's in North Yorkshire, you can see, and it's also in West Yorkshire. And then last year, for the first time, it's just popped over the border into Durham. How it got up there, nobody knows. We may never know. So here's the other big resistance focus that was around in that early map, and you can see it's still there. So our DNA is telling us it's really still down in Kent and East Sussex. It's pretty well established in London as well and spread up into East Anglia. We've got these odd spots getting out into Wiltshire, and one of Ewan Bates's Oswestry rats had it in. How it got there, again, heaven only knows. It was found in 68. Seems to be well established in London. Only the potent SGARs, those three that I mentioned, are fully effective against this strain. Difenacoum has some effectiveness, but only the three most potent ones are fully effective. Now here's the next one, and this really is a rather strange distribution. It's like someone's sort of fired a shotgun at the map, and it's everywhere and nowhere. Probably found in the early surveys, but we can't be sure because there wasn't a real focus of it. It's very strange distribution, possibly caused by multiple introductions or possibly caused by multiple mutations, spontaneous mutation events all over the place. It's very common in Europe, and it's the only mutation that's found at all in Germany and in Denmark. Again, only the most potent SGARs are fully effective. And as far as we can see, it could be pretty much anywhere. Now, here's the most severe one that we have. And this started, it was almost certainly present in that 1969 survey. It started on just a few farms in Hampshire and Berkshire. And I was working on those farms when we started to find these highly resistant rats, the L120Q resistance. It's resistant to all FGARs and to bromadiolone and difenacoum. So what this meant was that the ban on the use of potent SGARs outdoors really did cause the spread of this resistance, and it's now found all over central southern England. Almost any rat that we get from that area of the red splodges there, we find resistance. Very few susceptibles. We're now starting to see outlier foci increasing in number, and you can see by the red marks. The squares mean we found a homozygous animal, and the diagonal, the little diamonds mean that the animal was heterozygous. And where we're finding homozygous animals, that means that both mum and dad obviously had the resistance, and so it's pretty likely that there's a strong resistance hotspot in those places where we got the squares. Some French researchers got hold of this resistance strain in their laboratory, and they did some tests, and they found that it's got some of the animals have metabolic resistance as well. So they really are super rats. Now, what I want to do is just tell you about this study that we did. We couldn't do it for many years because of the issue with not being able to use brodifacoum outdoors. But we finally did 6 field trials in Hampshire a couple of years ago. 2 difenacoum 50 ppm, 2 bromadiolone 50 ppm, and 2 brodifacoum 25 ppm. And these trials really show you what was going on for 20 or 25 years in the area of this resistance. We did, so these are our field trials. We do pre-census census bait or pre-treatment census bait. And you can see down there in the 3rd column, the weight of bait that got eaten by these infestations in one day. So the infestations vary in size, 1.4 kilos, not very big, 10.6 kilos of census bait consumed in one day. We use some assumptions, from that we can get a rough estimate of the number of rats in the population. And you can see there's an estimate there, and they range from a low of 69 up to 520. It shows you the quantities of rodenticide used. And this is the amount of bait that we put down to try to control the infestations, and therefore the quantity in grams of the active substance we use. And then we do 2 independent checks of efficacy at the end of the treatment. And you can see the 2 difenacoum treatments. One estimate gave an estimate of 15% effectiveness, 15% mortality, but the other estimate told us the population had grown. Probably not much change in that. The other infestation, we got 6% control. Something very similar for the 2 bromadiolone treatments. In one, we used 43 kilos of bait against approximately 275 rats, and the population grew. So I want you to keep in mind, those 270 live rats at the end of this are living and breeding, and they're carrying 2.2 grams of active substance, or the equivalent of 43 kilos of bait in them, because the substance stays there, and they're available for consumption by predators. And that shows us what's been happening with regard to wildlife contamination in these areas where those 2 substances are used and still continue to be used. You can see the effectiveness of relatively small quantities of the brodifacoum bait, almost 100% wipe out in a very quick time. That 9.7 amount of bait is actually higher than really was used. We had to do some burrow baiting in that trial, and we couldn't get the uneaten bait back. So we clocked it all as eaten, but I'm quite sure it was a smaller amount. So coming to this latest development in this resistance story, and that's the development of something called hybrid resistance. And that's where a rat contains 2 of those different resistance mutations. It happened first in Scotland, and in 2016 we got a rat from Scotland just outside Edinburgh, and it had both the L128Q mutation that you would expect to see up there, but it also had the L120Q Hampshire mutation as well. Again, don't know how that happened, but that just one of those popped up, and we got nothing more. But really strangely, in 2020, we had a very small sample size because of COVID, And we only got 54 tails of rats, but 8 of those were hybrid resistance. And hopefully some of you, if you're looking on a reasonable device, you can see these little diamonds spread all over the country. There's one down in Dorset, there's one on the West Sussex, East Sussex border. There's one in Merseyside, some in Greater Manchester, West Yorkshire, East Yorkshire, Durham, and of course that original one up in Scotland. So how does this happen? We think it's what happens is individual foci of rats where all of the rats have the same mutation slowly get bigger and bigger, and they touch and they merge, and eventually rats containing different mutations meet and breed, and they would then pass both mutations to the offspring. And that's how we get hybrid resistance. We just don't know anything about the resistant status of these hybrid resistant rats at the moment. We've done no science on the effect of these hybrid resistances. So here's the whole story, and I won't go into this too much. Here's the whole picture: 331 tail tip samples we got over 5 years. Thank you so much. Many of you, hopefully some of you listening, would have sent those tails in. So you have helped us to generate this. We've got more mutations than any other country worldwide, but we've done more resistance testing more than anyone else worldwide. That sounds a bit like a COVID story more than a resistance story. Southern England is heavily resistant. It's been much sampled though, because we've been based in Reading University. We've been going out of the campus and touring around the countryside in central southern England, trapping rats, taking them back. And that's why we've built up this really detailed picture of what's going on. We've got so few samples from elsewhere, so we just literally don't know what's going on elsewhere. But you can see there's a big central area where we got almost no samples: the Midlands, Lincolnshire, Nottinghamshire, Derbyshire, all of those places, almost nothing. But where we have got samples, many of them are susceptible, which is interesting. Okay, so we need more samples, please. We need them from all of Scotland, all of Wales, and there's a list of counties there from which we've got almost no samples. So if you're listening to me this morning and you're sometimes trapping rats there, all you need to go online at CREW, not at RAG, at CREW. And there's a new CREW-funded collaboration with the Animal Plant Health Agency. Free rat and mouse tail testing. You can go online and you will be sent some of these kits. It's got an envelope not prepaid. Sorry for that. We send out so many of these things and don't get them back, so we weren't going to send prepaid. But everything else is free. All you need is stamp. Put the tail in the bag. It's already pre-addressed. There's a leaflet in there to tell you how to do it, and we will look at your tail and we will tell you what you've got, and you're here as quickly as we can possibly do it. So please send us loads more tails. So a little bit now about resistance management. What can you do? And really the golden rule, it's absolutely the golden rule of every resistance management for every pest species and every other resistance. Don't use things that the infestations are resistant to. So don't use anticoagulants to control infestations that are resistant to them. It wastes time and money and is a threat to public health. It causes the spread of resistance and increases severity by increasing homozygosity. It harms the environment when large amounts of ineffective anticoagulants, just think of those trials of difenicum bromidine in Hampshire that we conducted and I've just shown you. It harms the environment. Ineffective anticoagulants, masses are going into the environment and going into all of the barn owls and red kites that everyone is worried about. But obviously the question is, how can you folks know? So this is what has been happening. All of that Reading resistance data is going into a really super interactive program that sits on the Resistant Action Committee's website. Please keep in mind it's the committee, that's the International Committee based in Brussels. We feed them our UK data. They load it into a really clever mapping program, which is based on Google heat mapping. And you can home in on your home area, even your own house. You can tell this program how far you want to look outside. So you can dial the diameter of that circle. You can decide whether you want to look for rats or mice. You can decide whether you want to look for resistance or or susceptibility or both. You can turn on which strain you want to look at. And what pops into the box is it tells you all of the resistance that's found. In this case, it's within 93 kilometers of a spot somewhere to the northeast of Peterborough. And it tells you all of the different resistances that have been found. If susceptibility has been found, it tells you. And somewhere else on that site, it will tell you what to do. So it's a very handy resistance tool. And of course, so this is just another screenshot from it that you can see. Now, there's lots of spaces you can see where we don't have data. And if we could fill this map up so that pretty much anyone in the UK can drop a dot and we can tell them what's happening within 5 or 10 kilometers with a degree of accuracy, that would be just great. But we need more tails to be able to do that. Okay, coming to the end now, there's a new RAG, rodenticide resistance process, where to try to make things easier for people to work out what substances to use, we've issued a new grouping scheme. And you can see down the left side, there are 4 groups: anticoagulants, calciferols, narcotics, and gases. You can see anticoagulants has 3 subgroups, A, B, C. So any anticoagulant would fall into either 1A, 1B, 1C. You can see the groups, and these are the active substances that are available in the UK. Obviously, others are available elsewhere. Anticoagulants, these 3 anticoagulant groups, B contains bromodiolone and diphenhydramine, the less potent anticoagulants to which there is resistance in house mice and some Norway rats. And then the 3 brodifacoum, flocoumefen, for which we haven't got resistance either in house mice or in Norway rats. So all of those resistant strains I was telling you about are susceptible to those 3. But it gives you the recommended uses there. What we're hoping is that more people will begin to use this grouping and talk about these groupings, and it will then become the obvious thing as to which group you use depending on what resistance you've got. And we're hoping to talk to manufacturers and possibly to get these groupings put on the label. So when you buy something that's got bromodiolone in it, you'll see it's in group 1B, and you can look up group 1B and you can see the recommended uses of that active substance. So that's a new development that we're trying to help people to understand what's going on. So I think this is the last slide, practical resistance management. Just a few tips for you, please. There's some new RAG guidelines, both for rats and mice. We've just updated them. So go online at the RAG website and you can get those guidelines and have a look at them. You can find out about resistance in your area as much as we've got, so long as we've got some resistance information, by going to that Resistance Action Committee website. If there's no data, please do send tails. All the tails are welcome. So please go to the crew website, get some kits and send them off. Check the correct products to use against any resistant strains you may have. Only use fully effective rodenticide products. And if you're in central southern England, it's fair to say you can assume you've got L120Q. And if you're over in Kent and East Sussex, it's fair to say that we haven't found almost any rats that don't have Y139F. So you need to work on that basis. I think that's the end of the talk, and I'm hoping, well, that's taken just about half an hour, and I hope we got plenty of time for
Hi
questions.
there, Alan. Yeah, that's great, fantastic. Can you see me by any chance? Can you see both of us?
Okay. Yeah, I can now.
Fabulous.
Thank you.
So we've got 9 questions that have come through. People have been voting for their favourite questions. So yeah, we've got a few people that like this one. So I have submitted tails for resistance testing, but received no results or acknowledgement. Any idea how long this usually takes? So a bit of problems with sending them out. What's happened there?
Well, I'd need to know when this was done. We've just swapped from Reading to APHA, and you should receive, well, I would be hoping that when the tails are received, within a matter of days, you would receive an email to say that those tails have been received. There was a time with this process where we would, if there was an area where we got lots of tails before, and the tail came, say, right from the smack in the middle of that red blotch area that you saw on the map, we would say, oh, well, we haven't got much money. We won't look at that tail because we know it's going to be L120Q. Well, because we're now interested to look for hybrid resistance, we actually need to look for all of these. And so we are doing all tails that are sent to us, every single one. So all I can say is sorry for that. I will get into contact with APHA. If you want to drop me an email, and my email address is readily available, tell me who you are. I'll follow up specifically about that and get someone from APHA to get back to you.
Fantastic.
Should happen within days is the quick answer, and obviously
Yeah,
isn't.
no, excellent. That's great. Okay, I'm sure they'll be in touch. David Lodge here, he asks, or it's more of a comment and then a question. So we have a lot of issues with mouse resistance in London. Questions popped and disappeared. There it is. Would you say that in general there is a geographical correlation of incidence of resistance where L120Q is found and resistance in mice? Would be very interesting to have a presentation of, equivalent presentation on the house mouse.
No, I really don't think that there's a connection between the L120Q in rats and mouse resistance. We've only had about 100 mouse tails, and you might have seen I flipped over it quickly. In case I had time, I did put a mouse map right at the back of that presentation, but I took it down. We've only had 100 tails, so there's very little we can say. What we can say is that almost every tail we've had from London has had one or other of those resistance mutations, and some of the mice have had both mutations. So just like in normal rats, I was saying, we got hybrid resistance in house mice. There have been some reports of some resistance to one of those three more potent active substances. So some anecdotal reports of resistance to diphthiolone, and I've had 2 of those now, both from sources that I really trust. So I wouldn't be saying it on this if I didn't, if I hadn't heard from people whose comments I believe. So we don't know what the hybrid mice do in terms of the extent of their resistance, but I would say to David, hi David, good to hear from you. But I would definitely say it. We say to everyone, if you've got house mice, you must assume that they are resistant, and you would treat them as such. Go into that little graphic table that I showed you, and it'll tell you which substance to use against mice. And it doesn't. It suggests you don't use any first generations. You don't use bromidol and difenicum. You use some of those other groups. That's group 1C and I think 2 and 3.
Great. There's some more information. You put up a link, didn't you, to the relevant website? I stuck it in the discussion forum section as well. So hopefully everyone can have a further read on that if they need to, can't they, Alan? Great.
Okay.
So Steve here says, do you think that cholecalciferol use in the near future will be significant enough to have a detectable impact on the occurrence of resistance? And then in brackets, if it is no longer being selected for?
That's a crystal ball question, isn't it? It's a crystal ball question in about 3 different ways. Are people going to use it? Will it work? And will it have an effect to remove resistance foci? If people use it and if it works, it should be, and I know there's lots of data to show that it is fully effective against resistant rodents. So it should start to bring down the incidence of resistance. The problem we have is we start with a disaster. Really, resistance is so widespread. So if you begin to use, say, cholecalciferol in one area, you're surrounded, say, in central southern England by resistance. So you're going to get more resistant rats coming back in to populate that hole that you've created. So it's going to be a long time, I think, before we see those resistant areas getting substantially smaller. But who knows? It may happen.
Got any magic answers for now, have we? I suppose back in the 60 years ago, we probably couldn't have predicted the resistance, or maybe we could have done if if it had been brought up. But yeah, it's crystal ball stuff, as you say. Yeah. Great. We've still got a few minutes left. Good. Let's do some more. So we've got the interactive map you showed is great, but it would be even easier for technicians if there was a detailed map showing what anticoagulant rodenticide to use and where. Might that be possible at some point?
Well, I'm not. I think that's pretty detailed. You can dial in to a single building in that map, and you can ask the map to give you 1 kilometer, 5 kilometers, 10 kilometers around. It will tell you what resistant mutations are close by, and it'll tell you the distance to those mutations. And if you go into that table that I put up there, it will tell you what to use. So those are, yeah, those are not difficult things. I realize there's probably two things to do, but I was thinking, at least I was hoping, that that map was detailed enough to allow people to work out what was going on within a few miles. The problem that we have is we got blank patches. We got blank patches on the map. So if you go into say anywhere in Nottinghamshire, Derbyshire, Huntingdon, Bedford, Buckingham, and say what's around me, they'll say nothing, because we've got no tails, which is why I'm asking everyone who's got rats in those areas, please send us some tails.
Great, absolutely. Hopefully everybody listening today will take note and certainly get that done. So that's fabulous. As Ian here asked, are there UK forecasts in law to continue to restrict availability on farm for rodenticides to prevent resistant spread? Did I read that? And does that do you understand that how I read it, Alan? It's a bit of a bitty one.
I think I understand, and the simple answer is no. As far as I know, there's no intention to prevent on-farm use of any rodenticides that might be safe and effective.
Good.
Something might be going on, but I don't know. I don't know about it.
The short answer is the good ones, isn't it? It's quite clear. No, you're absolutely right. Terry here, how long does it take to get results back generally? You mentioned that before, didn't you?
Yes, well, it's a bit of how long is a piece of string, because what happens is they don't do these tails on a one-by-one basis. To run the extraction and the DNA sequencing, it goes through, it's a very expensive process. It actually costs about £100 a tail. And so they bank up 20 or 30 tails, then they turn on the machine and they run them through. So if lots of tails are coming in, the answers come back quite quickly. If somebody sends a tail and then nothing happens for a month, that tail is not going to be sequenced on its own. So I'm afraid it's difficult to predict. All I can say is the more tails we get, the faster it will be. And we certainly try to acknowledge the receipt of the tail so that you can be sure your tail has got through. And I'll be talking to our friends and colleagues at APHA to find out if that's the case.
We've got a minute and a half left. I think we should be able to get this in. So there's a question about behavioural resistance and how you think is best to face that type of resistance. And how do you know it's behavioural resistance and not technological mistakes that you've made? Yeah, I don't know if you can do it. It's now a minute, Alan. Sorry.
No, it's extraordinarily difficult for rats. What I would say is, in my experience, bait boxes are a big problem, and it's as much a resistance to bait boxes as it is to baits. And so if you can avoid using bait boxes by using equally safe and covered protected bait stations, we've shown scientifically that bait take from those covered and protected bait stations is way better than any bait station. Now, I know there are issues with technicians not using these bait stations. They may feel that the only safe way to put baits out is in a proper tamper-resistant bait station. I fully understand that, but so I haven't got a quick answer to that. It's a very difficult one.
It's a big discussion, isn't it? And we're always available at BPCA to help, through that discussion, as I'm sure, a lot of our exhibitors are as well. So again, before we get, the time is being extended for us, just for us to say thank you very much, Alan. That was fantastic. And your presentation will be ready for download once the event's done. So there's lots of people asking about it. So I think there'll be lots of downloads afterwards. So thank you very, very much.
Okay, yeah, I'm sorry if they were looking on phones. I suspect they didn't see all those little tiny dots in the maps, but I hope they will be later.
Fabulous. Thank you, Alan. And to all of the attendees, yeah, absolutely get to those exhibitor stands, have a chat to everybody. We'll be closing the show in the next 15 minutes. So if you don't manage to get it in, we will be here again tomorrow and Thursday. So again, thank you, Alan. Thank you, everybody that was managed to come along. Take care now. Bye-bye.
Thank you. Thanks, everyone. Bye. Bye-bye.